Hello, my name is Blake Porter, and in this video I'll be demonstrating the capabilities of my neuron models and what people can learn from them. To start with, there's two models here. They're both anatomically accurate and show the basic anatomy of neurons. So there's the soma, or the cell body. This is where DNA is housed and where signals coming into the neuron are processed. Coming off the soma are the dendrites. And dendrites are where signals come into the neuron. This may be signals from other neurons in the brain or from sensory systems like taste and touch. So coming down from the soma, going to this other neuron is called the axon. And axons are the part of the neuron that sends signals from one neuron to another neuron or to bodily systems such as organs and muscles. The core components of the models, though, is the physiology that they can show. So all of these dendrites have copper coils on them, and these copper coils act as touch inputs to the neurons. So neurons primarily work off electricity in the form of ions, charged particles, usually sodium and potassium. And when neurons get an input, these ions flow into the neuron through different channels. And these ions cause the voltage inside the cell to change. And that's what the LEDs here are reflecting. So the LED brightness reflects the voltage inside the cell as it becomes more positive with more input. You'll notice when I let go that the LEDs go dim and then shut off again. And this is the cell going back to its resting membrane potential. It likes to hang out at about -70 millivolts. This is the resting membrane potential of the neuron, and there's a bunch of different homeostatic mechanisms that will force ions out of the neuron to bring it back down to its resting voltage. But if we give the neurons enough input, the soma will process that input, and there's an LED in here which, as it hits peak brightness, that reflects what we call the action potential threshold. And once the soma LED hits that action potential threshold, it will fire an action potential down the axon to this other neuron, as you can see there. And there's two key ways to get a neuron to fire, which we call spatial summation and temporal summation. So spatial summation is when inputs are coming into the neuron at different parts spatially. So perhaps just one input is coming in, and as you can see that's quite weak. It takes a very long time for the signal to travel down the dendrite to the soma. But with spatial summation, if the neuron receives input to two different spatial locations, that input is summed and the signal travels much faster down the dendrite. The soma processes it and it sends off an action potential. The other way is temporal summation. So temporal summation is inputs are summed temporally in time. So if inputs are not too regular, they're not coming in too fast, then nothing much happens. The signal's not strong enough to reach the soma. But as those inputs are summed, as more and more inputs come in faster and they're summed together, it's enough to get the voltage inside the cell to rise and reach the action potential threshold. You may have noticed that after the neuron fired an action potential, this red LED flashes inside the soma. And this red LED reflects the absolute refractory period. And this is a time right after the neuron fires an action potential, which it is no longer able to fire an action potential transiently. During this time, those channels that allow ions to flow into the neurons are being reset, so no matter how much input the neuron gets, it won't be able to fire an action potential. So you'll see, even though I'm holding this down and giving it a constant input, there's a short period of time after the action potential where nothing happens. This is the absolute refractory period. So that's the key physiology that a single neuron can show, but these two neurons are actually functionally coupled here at what we call a synapse. So the axon of this neuron is connecting to the dendrite of this neuron through this connection here, which is a synapse. The red LED in there, which is actually an RGB LED, is showing us the strength of the synapse. So right now it's very weak, it's red. When this neuron fires, not much happens over here. Before we get into how synapses work, just a bit of nomenclature. This neuron is called the presynaptic neuron. This neuron is the postsynaptic neuron. So in order to change this synapse, which is called neuroplasticity, we can either strengthen it or weaken it. So to strengthen it, very specific conditions must be met. That is, the presynaptic neuron fires first, followed shortly after by the postsynaptic neuron. And if we do that, if we do these pairings, pre-post, this synapse will get stronger. And the color of the LED in the synapse will reflect the strength. So it'll go from red to blue to green. So I'll do a few pairings now. Pre-post, you notice that LED will turn a more purple color and transitions over to blue. And as the synapse gets stronger, the connection between the two neurons is strengthening. So the presynaptic neuron over here will begin to have more influence on the postsynaptic neuron. So if I just take a break for a second, you'll notice that this current is spreading out throughout the whole dendrite now. Because as this synapse gets stronger, more channels, more ion channels open up in the synapse. This lets more ions flow in, and that's how this neuron will have more influence over here, and why strengthening synapses are important. So with a few more pairings, the synapse will go green. This reflects the strongest synapse. And the synapse will be so strong that the presynaptic neuron can get the postsynaptic neuron to fire without any more help from me, as you can see there. So that process of strengthening the synapse is called long-term potentiation. The synapse is being potentiated, it's being strengthened. But we can also weaken the synapse, and this is called long-term depression. So I'll go through that now. There's quite a few ways to induce long-term depression. One is simply that the postsynaptic neuron fires without any input from the presynaptic neuron. So if I just make the postsynaptic neuron fire, you'll notice the synapse begins to become more blue as it weakens. And there's a few other ways to do long-term depression. One is the presynaptic neuron fires first, followed by the postsynaptic neuron, but the postsynaptic neuron takes too long to fire after the presynaptic signal arrives to it. So the presynaptic neuron may fire first, but if the postsynaptic neuron takes too long to fire, then long-term depression will be induced at the synapse. Thinking that because the postsynaptic neuron was able to fire and the presynaptic neuron input came so long before it, this input probably wasn't that important. The other way to induce long-term depression is if that the postsynaptic neuron simply fires first before the presynaptic input reaches it. So we can get the presynaptic neuron to fire, but if the postsynaptic neuron fires first, long-term depression is induced. And eventually we can get the synapse to go back down to red. And you'll notice again, when it's red, barely any current is flowing into the postsynaptic neuron. So those are sort of the core physiology demonstrations that the neurons can do. I hope you learned something and enjoyed it. Thanks.